Hey guys, we are going to finish up talking about the ear, hearing, and balance today, and we're going to talk more about the physiology of hearing and equilibrium or balance. So we're going to start with hearing. I know I'm blocking out a little bit of the text at the bottom, but you guys have these slides shared with you, so follow along as we go. So we're going to start with the organ of Corti. This is located within the cochlea, and the receptors here that we talked about, the mechanoreceptors, are going to be hair cells that are on the basilar membrane. And then above the hair cells, you have a gel-like membrane called the tectorial membrane, and this is actually capable of bending those hair cells. Those hair cells are attached to the cochlear nerve, and so when they're bent, it's going to transmit nerve impulses to the auditory cortex on the temporal lobe. So if you have the cochlea, which again, it looks kind of like a snail, and you were to take a cross section and look inside, what you see are the three different chambers. So you have the vestibular canal, the tympanic canal, and then you have the cochlear duct. Sort of contained within that cochlear duct, you have the organ of Corti. It's got the basilar membrane, and then it's got the hair cells attached, and on the top of that, it's got the tectorial membrane. And so as vibrations in the fluid move through the cochlea, they move the tectorial membrane. When this moves, it causes the hair cells to bend, and then a signal or impulse is sent down the cochlear nerve to the auditory cortex. This is just some more diagrams, again showing you the same thing. So here is the cochlea. We've got another cross section. So we are looking again at the organ of Corti here in the middle, and then this is just a very zoomed in version of the organ of Corti. So again, you've got the basilar membrane, you've got the hair cells, the tectorial membrane. When this moves, it moves the hair cells or bends them. When they are bent, an impulse is sent down the nerve fiber. So, vibrations from sound waves move the tectorial membrane. Keep in mind that this is all starting from the external ear. So you've got your pinna, which is kind of shaped like a funnel to collect noise or sound. Those sound waves are then sent down the external auditory canal. They hit the tympanic membrane. They cause vibrations there. The vibrations are then sent through the middle ear via the ossicles, the three bones that we talked about. And then the vibrations are sent to the cochlea. So as the vibrations from sound waves move through the cochlea and they move the tectorial membrane, the hair cells are bent, an action potential starts in the cochlear nerve, and then it's continued and sent to the auditory cortex. This is just, again, two diagrams. This is nice because it's showing you the different parts of the ear and how they're involved. So again, you have the pinna to kind of help funnel sound in, that the sound waves are going to move down the auditory canal to the eardrum or the tympanic membrane. Once the vibrations hit that, they're sent to the hammer, anvil, stirrup. And here you can see that sound is amplified, so you can see the wave gets bigger. Then it goes to the oval window, which you can see right here is like the opening to the cochlea, and sound is going to travel through the fluids in the cochlear canal. So here you've got the sound traveling through the fluids, and it stimulates the organ of Corti as that happens. So the basilar membrane is here, and so this is essentially where the signals are picked up and then sent to the cochlear nerve and then to the brain. So now we're going to move on to equilibrium, and we're going to talk about the receptor cells and where they are located. And so we've got the vestibule and the semicircular canals, and because we've got two different structures, we actually have two different kinds of equilibrium that we're going to talk about. So there are two functional parts. You have static equilibrium, and then you have dynamic equilibrium. Static equilibrium, you are going to have macular receptors in the vestibule, and they're reporting on the position of the head. And so they are going to send information via the vestibular nerve to the brain. The macula are hair cells that are embedded in an otolithic membrane. And then on top of the otolithic membrane, you have something called otoliths. They look like tiny stones, and they're floating around in sort of a gel around the hair cells. And so when your head moves, it will cause the otoliths to move due to gravity. When the otolith moves, the otolithic membrane moves, and again, it causes a bend in the hair cells. So here you have a head upright, and And this is sort of what the anatomy looks like at that point. So again, you have the macula with the hair cells. On top of that, you have the otolithic membrane, and then sitting on top of the otolithic membrane, you have these otoliths, or like these tiny stones. And so when the head is upright, everything else is upright. Now, when you tilt the head down or back or to the side, what happens is the force of gravity is going to pull the otoliths, and then that's going to pull the otolithic membrane, and then that's going to bend the hair cells. And so again, when those hair cells bend, you have signals that are sent via the nerve fibers. Okay, and this time it's going to be the vestibular nerve. With dynamic equilibrium, here we're looking at crista ampullaris, and these are receptors in the semicircular canal. So with static equilibrium, we're talking about the vestibule. Dynamic equilibrium, we're looking at the crista ampullaris. And so these are receptors, again, they are tufts of hair cells. So again, I know I'm covering this a little bit, but this would be the crista ampullaris. You have the hair cells sort of embedded within that, and then on top of it you have this gelatinous cap called the cupula, and this is going to cover the hair cells. Now, so if you were to, let's say, walk this way, as you're walking, the endolymph inside the semicircular canals is going to flow backwards. It's going to push on the cupula. That is again going to bend the hair cells. That is going to send a signal through the nerve fibers. So whichever way you are walking, the force of the endolymph is going to push the cupula in the opposite direction. Think about sort of like accelerating in a car. As the car accelerates forward, you get pulled back a little bit. So the same sort of thing happens with the endolymph. As you move one way, the endolymph moves the other way. It's going to cause the cupula to bend the hair cells, and it's going to send a signal to the brain so that you can sort of interpret your position and your movement. So this is action of angular head movement. And again, the cupula is going to stimulate the hair cells for that angular head movement. An impulse, again, is sent via the vestibular nerve to the cerebellum. And that is it. So I'm going to resend these slides to you guys so that you can follow along and take notes as you watch the video, and I hope this helps. All right, bye guys.